All right, guys. So, welcome back. The good news is that we covered so much material in this that I didn't realize that I was ahead of myself. So, take a deep breath. We are actually moving at a really good speed. So, this is week 1, and we've already kind of covered most of the stuff for week 2 as well. So, we are moving at a really good pace. So, take a deep breath. Next week, week 2, should be a little bit lighter. Again, reaching out to me if you have any trouble. Some of you are like, this is cake. Others of you, let me just turn this microphone. I can see it flopping around. We at least want it flopping towards my face so I'm not cutting in and out. So those of you that this is a breeze, great. I'm glad. For those of you that might be struggling, that might think that this is a big topic, remember, I had to learn this in less than 8 weeks. So we've got this. Don't panic. Reach out if you need any help, okay? All right. So last time we talked about Mendel and Mendelian genetics. You can also correlate Mendelian genetics with transmission genetics. It's how traits, phenotypes are passed from one generation to another, okay? And we talked about pedigrees, how that moves through family generations, how we can see, oh, no. Did we talk about that yet? No, that's what we're talking about now. So we're about to talk about pedigrees. So we talked about the postulates, the 4 postulates, the unit factors, which equal genes, the dominant versus recessive, which we talked about. Those are just basically, is it functional protein or non-functional protein that's allowing it to express. We talked about the laws of segregation, so the coin flip. And then we talked about independent assortment. So we started to look at probabilities within that. So now, and we did talk about the chi-square analysis or the chi-square analysis. I'm not really sure how to say that. Okay, I'm teaching myself. Okay. What we're looking at is the expected versus the observed. So when we run an experiment, and Mendel said, this is what we expect, 75-25. We expect a 3-to-1 ratio. This is the outcome that we expect. But if we don't get that and we get pretty close to it, this is what we actually observed. Is there something funny going on? Or is this just that there's no real difference? Which when it's that close, there's just maybe some flukes or some environmental things, right? Okay, so erasing that, we are now moving forward. So remembering that this was chapters 1 through 4, okay? Section 2 is 3. I had it down. It's in the syllabus. But we are ahead of the game, okay? We are ahead of the game. Don't let yourself slow down. Like, keep up this good pace, but here we go. I'm excited because it means little concepts that maybe I was struggling with or something, I have a little bit more time to fill in with those. So maybe that's something that you guys feel the same way about. Okay, so pedigree analysis. This is the basics. So when we're talking about pedigrees, it's almost like detective work. So let's spell this out. Pedigree analysis. It's like our detective work. So like Clue. Who killed who in what room with what weapon? Who passed on what trait? Does it skip? So the trait is what we're looking for, okay? So basically this is looking at family trees, and family trees will show us the inheritance of certain traits across generations, okay? So when we're looking at this, when you look at these trees, we can look at a circle would be a female. And a square is a male. Okay? Then we can look at a line. This is like reproduced, or let me give the real word. So this is a horizontal line equals mating. That's the word they use in your book. And then we have vertical line equals offspring. So basically, this is just drawing a family tree. Now, when we are looking, was somebody affected by something, to look at it and see if it was affected, we would show square or circle shaded in. That means they got the trait or they got the disease, whatever that might be. And so when we're drawing up a pedigree or a family tree, that is what we're looking for. Okay? We're looking for, did they get it or did they not? And it shows how these genes express. Do it? They skip a generation. Okay? So the first one that we have is autosomal dominant. So with the rules from Mendel, we know that they're going to get it. It's dominant. If the trait is there, if the gene is there, if the allele is there, they're going to get it. And what autosomal means, it does not skip a generation. So when we look at this, trait appears in every generation. In every generation. Okay. Another thing is both male and female are affected the same. Okay. One more. It is, we have our example, Huntington's. Huntington's disease. And this is a disease of the nervous system, okay, where it starts to impact movement. So they have really unbalanced or uncoordinated movements, some shaking. If this gene is present, then it will pass on, okay? So this is where they can go to looking at the ethics behind all of this genetic stuff. Is we can look at a family and we can say, well, if they are autosomal dominant for this and they have that trait, should they reproduce? That's kind of what we're talking about when we're looking at all of this gene stuff. We can now determine, okay, well, they have this gene for Huntington disease, but remembering that the environment also allows it to pop up. So the second one is autosomal recessive. I'm just going to put auto recessive just to save time. Okay. Auto recessive. So this skips generations. Okay. This skips the generations. And you guys in your lab, there will be like a video more on this. So you guys can see like what this actually looks like. But you're just saying like, if somebody's has the trait, like they have the gene, right? They're married and they reproduce. It's this one, it's going to show up within their genes, right? So female or it's going, the trait is going to be expressed. Whereas here, maybe they have the trait, like they have it within their genes. Their kids don't get it, skips a generation, but then their child gets it. Let me make sure. Yeah, both males and females are affected the same. So everything is the same here. It's just, both parents have to be carriers. So they are carriers of the trait. So carrier and a carrier of the trait. Okay, so they're both carriers. So let's say R. There's our trait. It's recessive, passed on, skips generation. Okay? And here, R. Both parents have to be carriers, right? Because it skips a generation. Okay. And like I said, in the lab, it will go into this into a little bit more depth. Okay. So it skips a generation. That's the big thing that you need to know about autosomal recessive. So this is something like cystic fibrosis. And the pedigree is just literally seeing like, okay, if we can trace this, if we can follow this, one, we can follow it down, and two, we can follow it back up and see where it came from. The next one is X here. Let me erase. The next one is X-linked. So that obviously means it's related to females. So the mother is the carrier. And they generally pass it to sons, right? It's more likely to affect males. Okay, so X-linked is males affected, like I said. It comes from the mother, right? Because mom has two XX, so the gene is on the X, XY. Males are more likely to get it. Okay. Let's see. It can skip a generation. Can skip. Sons are carriers, the moms are the givers. And here's an example of hemophilia. So hemophilia is a disorder where cystic fibrosis is where there's like mucus buildup in the lungs and in the intestines. So it gets progressively harder and harder for them to breathe. It gets progressively hard to pull nutrients in. Hemophilia is where they don't clot well. So their blood, they have a clotting disorder. And so they bruise easily. If they get any kind of wound, they bleed out really, really easily. So that's something to watch out with that. Okay. And it's more common in males. Here's the other one. This is X-linked recessive, by the way. Let me put that. And then we'll have X-linked dominant. Hopefully this is making sense with you guys. X-linked dominant would be both males and females, but females more. But more likely in females. Okay. The fathers pass it to the daughter. Okay. That's how it gets passed on. Okay. And then it does not skip generations. No skipping. So this is something like Fragile X syndrome. Okay. Forgot that E. Okay. So that's the difference between, so you can see dominant doesn't skip, can skip. Okay. And then we have Y-link. It only affects males, right? It's very, very rare. And it's passed father to son. So we've talked about X-linked. We've talked about the Y-linked. Is males are affected, it's passed fathers to son, and it is very, very, very rare. So rare, I don't even know if I remember a syndrome or a disorder that's passed that way. Okay. So basically, like I said, pedigree analysis is the detective work. It's like the game Clue. And these are some questions that I thought of. So who inherited it? How did they inherit it? Does it skip? And does it favor one sex? Those are all questions that these pedigree analysis ask. And you can think of it just like a Clue game, like the game of Clue. Easy peasy, right? Okay, so we talked about Mendel, and he made the game before the pieces were even discovered. So he was making the rules before the game had even begun. Okay, he didn't know what he was doing. So when we're looking at that, and we've advanced in technology, we've advanced in our research, we have found some exceptions to the rules, right? Of course. Just like I before E except after C, which is ridiculous. Okay. So these are the exceptions. Let me spell correctly. Exceptions. Okay. These are the exceptions. So 1, incomplete dominance. All right. What this is, it's not to the 3-to-1 ratio. It's 1-to-2-to-1. Okay. So maybe we get, it's easiest to look at this in flowers. So maybe we get 1 red, 1 white, but something weird happens and then we end up with some pink ones. So incomplete dominance. There's an intermediate phenotype. So this shows up as the intermediate phenotype. Okay. We have codominance. So this is like red blood cells, the blood typing, A, B, O. Okay. both alleles expressed at the same time. Expressed. Sorry, let me get that thought out. So this would be AB blood. They have AB blood. Okay? Multiple alleles. I'm just going to put this right here. Okay? So more than 2 alleles exist for a gene in a population. Again, this is our A, B, O. More than 1 or 2 alleles. More than 2 alleles. And what this allows for is continued patterns, continued genetic diversity, or expanded. I think your book likes the word expanded patterns. Remember, genetics is just patterns, okay? They also found something called lethal alleles. Oh, 4. Okay, lethal alleles. Some of the codons they found cause death. Codons will trigger death. Okay, simple enough, right? We don't need to elaborate on that. There's some codons that cause death, some combos of alleles that just mute it. So it was, in your book, it talks about an experiment where the mice had yellow hair. If that was expressed, it would just, they would die. Okay. Oh, I don't know what I was writing there. Epistasis. Okay, epistasis. This is where one gene masks another one. So this is, you could look at this in dogs, labs. So labs. One gene masks the other. So we can look at this as like, for the coat in labs, they could have black or brown, and they're automatically going to get the brown, but that this is the color and then the pigment deposition. So our ratios get modified. So we can have the color or the pigment deposition. Okay, no pigment. So it looks like this. B black, B brown. It could look like E, the pigment deposition. So I'm going to put deposit. Maybe if I could spell. And then E is none, no deposit. Okay, no depositing pigment. Okay, so this will modify our ratios automatically. Okay, most of the time if black's there, they're going to turn black. Okay, 6, pleiotropy. I'm making sure I spell this right. This one is a hard one to spell. Don't judge me, guys. One gene can influence multiple traits. So this is like in Marfan syndrome. So in Marfan's, it can, not only can it affect like their heart, but it can affect their ligaments and their tendons as well. So one gene is affecting heart, ligaments, things like that. Okay. All right. I didn't even write anything on number 6 except the name of it, but that's okay. These are just the exceptions, right? Exceptions to the rules. Okay. I think we have one more or 2 more maybe. Okay, yeah, 2 more. Okay, polygenic inheritance. Polygenic inheritance. And this is many genes contribute to, let me spell that word out. Many genes contribute to a single trait. Many genes equal one trait. And that would be like looking at human skin color or human height. Like the heights of humans vary, the heights of human skin colors, they can be impacted by multiple genes. Okay, and then the last one, and this is really big, and this is actually a new, big, and upcoming area of study, is the environment. So gene-environment interaction, They used to think that there was nothing involved with that the environment did not impact genetics. But now what they're finding even is that even before they're born, so epigenetics. And that's a whole fun topic, and we'll try to dig into that a little bit as well throughout this. But those are the exceptions to the rules. I'm going to say them one more time again so that we have them all together. Just real fast. Exceptions. Incomplete dominance. Red and white are crossed, they get a pink. There's an intermediate phenotype. Codominance. Both alleles are expressed fully in the heterozygote, AB blood. Number 3, multiple alleles. More than 2 alleles exist for a gene in a population. That's the ABO blood group. So we're expanding the patterns. Lethal alleles. Some combos of genes or some combos of alleles cause death. Epistasis. One gene masks or modifies the effect of another. So they're impacting each other. So for coat color in labs, gene 1 versus gene 2. Gene 1 is black-brown. Gene 2, pigment, no pigment. They impact each other, okay? That modifies the ratios. Pleiotropy. One gene influences multiple traits. So that's looking at things like Marfan syndrome, okay? Polygenic inheritance. Many genes contribute to a single trait. So we're constantly getting variation. That's human height, human skin color. I think even eye color has something to do with that as well. And then genes. The environment impacts our genes. And we'll even dive more into that with epigenetics. Okay, so the last thing that I want to do is I want to look at Mendelian genetics versus the modern view of it. Okay? So his laws and the modern view. Okay, we kind of already did that last time, but we're going to do it one more time. So we're going to put Mendel here. Mendelian genetics, or we could put in here, just for you guys to have it, transmission genetics, okay? And then we have extension, or the exceptions. I already did the modern. So this is the comparison of Mendelian laws and the exceptions to those laws. Okay, so if we just sit here and we look at the dominance, we can look and we can see that Mendel said there's a dominant allele and a recessive one, right? That's just his law. If we look at the exceptions, we can see that incomplete dominance, and we can see that codominance are the exceptions. Codominant, incomplete, and codominance are the exceptions. So again, remembering incomplete was the intermediate phenotype, right? And codominance was AB blood typing. Yes? Okay. So we look at alleles. Okay. Each gene has 2 alleles. That's what Mendel says. Whoa. Did y'all see that? Each gene. That's where I'm at, guys. Each gene has 2 alleles. Okay? But what we found over here, the exception to that rule, is multiple alleles exist in each population. So that was the height and the skin color. Okay. Right? No. Multiple genes would be epistasis. No, multiple alleles. ABO, blood typing. My bad. Sorry, guys. ABO. This is the AB, where they're both there. And this is the pink, or intermediate phenotype. Okay. Let's go back to, so we looked at alleles. I'm passing back and forth. Ratios. So he said 3 to 1 phenotype, right? And 1 to 2 to 1 genotype. Right, that was when we looked at our chart, T, 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 and, oh wait, this is a little t. Okay, so 1 to 2 to 1. Those are the genotypes, right? Just reminding you. And the phenotypes was that 3 of those are tall, 1 of them is short. Okay. Ratios. Now, the exception is that they found some combos are lethal alleles, and they will cause death, and so they will mess with the ratios. Okay. Independence. So independent assortment. Okay. Genes assort individually, right? Well, the exception was epistasis, saying that some genes mask the effects of others. So again, that messes with the independence, that they work independently of each other. One gene equals one trait. Each gene affects one characteristic. Right? And we found that in pleiotropy, the exception, one gene can affect many traits. Okay? And that we found was in Marfan's. Okay? One gene equals one trait. Okay. Traits are discrete. These are things that Mendel said, right? These are the laws. Okay? Well, tall versus short, that's an example of that. We can look at that and we can see polygenic inheritance. That is looking at many genes add together to make continuous variation. So many genes coming together to add variation. And that variation is continuous. Okay. The last one is genotype and phenotype. You guys can see this. Phenotype. Okay. Environment has no impact. No impact. And we talked about that. We could just put epigenetics right here. Okay, but also the genetics of your, or the environment of your lifetime will impact it. And so they'll say it's all about your genes. Really, what they're coming to find is that it's actually 95% more about your environment than actually about the genes that you have, right? Because there can be non-functional proteins that these genes are expressing, or they're locked up real tight. And you'll see in the structure of DNA what is occurring when we have to make copies of it, okay? So let's sum this up, all of this information up, so that you guys know kind of the big overview of this and then where we're headed. Okay, so Mendel, we talked about him. We actually went through the timeline of genetics. So we kind of went through the heart of genetics, how it all developed, okay, and where we're at now. Mendel, he was the pioneer. He came up with the 4 postulates, the unit factors, the dominant recessive, the segregation, the laws of independent assortment. So he came up, he made the pieces, the rules of the game before the pieces were even developed. Then what we did was we linked throughout this throughout history, we linked chromosomes and DNA to this and genes, and we were able to dig into it more. We saw how mitosis and meiosis are affected and how gene expression is affected by meiosis. We talked about probabilities and how probabilities underlie all inheritance, remembering that genetics is just patterns and predictions, okay? We talked about pedigree analysis, which is family trees and how traits pass down the line. So we're looking at it as like the game of Clue, like who gave what trait, did it skip? It's looking at it like it's a game of Clue, okay? And then we looked at the extensions and the exceptions to the rules. So really, we kind of logically went through molecular genetics. And now what we're going to do is we are going to get to the machinery behind it. So what we're about to progress through next video or next week is the machinery behind it. So we're going to be talking about DNA more, the structure of it, transmission, transmission, transcription, all of those fun things that maybe some of you might be getting a panic attack, but really it's not so bad. I did teach this part of genetics last year and it's actually quite fun. Okay, so don't panic, take a deep breath. You just went through the logic of it. Now we're going to start getting into the dance that happens within the cell when all of this is happening. Okay? All right. I look forward to your questions, your comments, your corrections, if there is any. And we will talk soon.